Live. Okay. All right. Thank you everyone for being here and for coming. I'm going to give you a little bit of an update in the work that other people in the lab, other than me, have been doing on Duchenne muscular dystrophy in particular. As you know, we work on a few topics, but this is the work that folks have been working on Duchenne's and focused on largely two papers that we're working on sending out at the moment, and I want you presenting later in the semester, right? No, okay. I have a little bit of a update on Mackenzie, but I may or not get to it at the end. So see if I can. Right. Okay. I don't know how this works. Working. Okay. All right, so first of all, I'm going to start by doing a small primer on muscle, just in case you are not familiar with how muscles work, and then a little bit about Duchenne's, and try to make the case of why we use worms to study this disease, and then get to the protein that is at the heart of the disorder, and then some of the work that we've been doing to try to understand the disease. And if I have time, I'll get to some future directions. All right. So every movement that you have ever made and you will ever make in your entire life, you have to thank muscles for this. These are contractile cells that are beautiful and very well organized inside. And in my physiology class, I tell students, it's like looking at a Harley-Davidson, an engine. You look inside, and everything inside of the cell is there to facilitate the production of movement and power, right? And so things aren't just lying around, they are there for a particular reason. And so when you go to move, your muscles typically are connected, your striated muscles at least, are connected to your skeleton, and they shorten and they generate tension that can then be used to move your skeleton. And at the heart of that is this contractile fibers, this arrangement of the sarcomere, where you have the thick filament, myosin and the actin, and they basically slide past each other, as you see in this diagram. And that sliding is what generates the force. The force then travels lengthwise in the muscle, so the entire muscle shortens. However, a lot of that force doesn't just travel lengthwise along the axis of the muscle, it also goes in other directions, therefore generating forces that can transfer to other parts of the cell that the cells need to take care of. Because if you think about it, cell membranes are lipid bilayers. They are not much more stronger than a bubble of soap, so very fragile. Yet these muscles, these cells are able to generate forces that allow you to lift couches and do all kinds of feats, right? And so something needs to be in place to protect cells from the forces that they themselves generate. Another piece of the puzzle, and I'm not going to go into all the details. You can take my animal phys class if you want to learn more about this. But something that is very important is that calcium plays a very important role in the generation of muscle contractions. All right. And so in this picture here, you have a motor neuron arriving from your spinal cord. This might be your brain telling your pinky to wave at the audience. In order for that to happen, the motor neuron needs to excite a muscle cell. And at the neuromuscular junction, which is this connection, the excitation is transferred from the neuron to the muscle. And then that excitation travels deep into the cell. And through a trapdoor kind of linking of two channels, you have this ryanodine receptor that opens the gates for calcium, which in your muscles is being stored inside the endoplasmic reticulum, kind of like a cistern. And when this excitation arrives here and these gates open, calcium floods the inside of the cell and binds to the complex here, the sarcomere, allowing the fibers to slide past each other. Of course, this is happening every time you move anything in your body. But for that to stop, this is how you take one step But if you want to take a second step, you need to relax that muscle first. And so equally important to releasing this calcium is to get rid of it once that muscle needs to relax, right? And to accomplish that, the renin receptor does not play a role, but you have a calcium pump here, the Sarca pump, which is a sarcoplasmic reticulum calcium pump, and that kind of pumps the calcium back into the ER so that the muscle can now relax, okay? There are other sources of calcium. Some calcium can come from the outside of the cell, and that definitely plays a role in smooth muscle and cardiac muscle, but in striated muscle. But largely, the calcium is coming from this avenue. And this is important to us because when during disease, this calcium can become dysregulated, and you might end up with too much of it lying around, and that is not a good thing. If there is any calcium lying around, that muscle will be contracted. Okay, and so we don't want that under normal conditions. So the forces that this muscle generates, some of it is transferred to the skeleton, so you can actually move. But some of it, as I mentioned, kind of is diffused outwards towards the membrane. And so you need something in place there to kind of protect the membrane from that. And this is where our protein comes in, dystrophin, which plays a crucial role in absorbing some of these lateral forces that are happening in the muscle. So dystrophin is a large protein. It's part of a complex called the dystroglycan complex. This is the rest of the complex here, these other proteins. And here you see dystrophin. They have these spectrum repeats, which are like tiny springs. And in this paper, people actually use tweezers to pull on this dystrophin and then measure the force that it requires to unfold each of those spectrum repeats. So the idea is that this is like a shock absorber, kind of like when you're driving down the road and you go over a puddle and this thing absorbs some of that energy so that you don't, right? In addition to this force, dystrophin also plays an important role, turns out, as a signaling molecule. And a lot of the players here kind of use dystrophin to find their way to where they need to be. A lot of channels, ion channels also need to go there. And so it's not just structural, the role of dystrophin, but also functional. So that is kind of how muscles are set up. But what happens during Duchenne muscular dystrophy? So first of all, I'll tell you a little bit about it. It's one of the most common muscular disorders. It's 1 in 5,000 males. The dystrophin gene is in the X chromosome. So males, we only have one. And so if you have a mutation on that gene that takes the protein out, then you develop this disorder. It's much rarer in females, although it does happen. It's a relentless disease. You know, kids start showing this, symptoms by the age of 3 or so. They're usually in wheelchairs by 10. They will die of asphyxia. They suffocate to death in their sleep because the diaphragm is a muscle, and then without ventilators, they will die. But thanks to ventilators, they can live a lot longer because of that. But eventually, they will die in their 30s, typically because of cardiac failure. Okay. So the gene itself is huge. It's the second largest gene, 250,000 bases. Most genes are 10,000 or something like that. It's a complex gene. It has many isoforms. This means that the same gene can give rise to many different versions of the protein, and they are expressed in different tissues. And we'll talk about that a little bit, which complicates, the matter more in the terms of the disease. In terms of how the disease affects different muscles, in terms of striated muscle, it leads to atrophy, which leads to loss of ambulation. That means kids need to go on wheelchairs. They cannot move on their own anymore, and problems with ventilation, respiration, as I mentioned. And cardiac muscles have also a host of problems. Cardiomyopathy is one of them, but also additional ones. And this is ultimately the cause of death for most patients. Okay. Much more understudied is the effect of the disease on smooth muscles. 75% of patients have severe GI impairments that require clinical intervention. All right, and so this is something, constipation and lots of problems in the bladder and the GI tract, that leads to a worsening of the quality of life for the patients of the disease. And in addition, it also has a lot of neurological aspects to it. There's reduced IQ. The gene is expressed throughout the nervous system, and it does have effects there. They are non-lethal, so they are typically understudied by comparison to other to the muscular side of the disease. But this is something that Mackenzie studied in our lab right now. And as I mentioned, there is no cure. Okay. In terms of the pathophysiology, we understand little about it. We have kind of glimpses throughout the progression of the disease. We do know that there is an increase in the levels of calcium in the muscles of these persons. How the calcium gets elevated is not entirely understood. The idea, one of the main ideas, is that calcium enters from the extracellular space when the muscles generate lesions on the membrane, and so the calcium can flood in. But there is also the possibility that signaling problems might lead to an increase in calcium, and we'll talk about that. Eventually, that increase in calcium, which is buffered by the mitochondria, and kind of the mitochondria help take care of that a little bit. Eventually, that leads to mitochondrial stress, overload, and eventually mitochondrial failure, which is typically the last thing that happened to those muscles before they die, all right? So the muscles undergo damage, they undergo necrosis, and throughout the entire disease, there is the presence of chronic inflammation as these muscles are suffering throughout the body of the patient. And so in this case, inflammation usually is a good thing, but not when every muscle in your body is undergoing inflammation, right? And not when it lasts years and years. And so when the muscles disappear and die, they are replaced by fibrotic tissue. Here's some pictures of human healthy cross-section of human muscle here. You can see the different fibers, and here is dystrophic, where you can see that there is the absence of tissue there. Okay, in terms of treatments of the disease, primarily corticosteroids, because they're anti-inflammatory, and because this is such a huge load on the patients. That is the main treatment right now. It helps pretty much all patients, regardless of what mutation they have, but it has many side effects. It also loses potency with time, and so it can only be given for a period of time. It kind of expands the life of the patient for about 10 years, but it's not a remedy. These patients are still in wheelchairs. They're still, very much impacted, just at a slower or at a lesser level for a few extra years. So with corticosteroids, maybe, instead of dying in their early 30s, they make it to 40s, but, not much more than that. There are additional treatments, but they are all bad, basically, right? Like there is, people try to deliver small versions of the dystrophin protein to the patients. And the problem is that even though you can see the small versions of dystrophin in there, there is a problem with resistant immunity. These things are packaging viruses. And if you're immune to this virus, then you can use the treatment. You can only get treated once, then you become immune to the virus and you cannot have treatment again. And also there is not really huge improvement as a consequence of the treatment. The same thing, there is exon skipping where like constructs are delivered that basically target the mutation so that when transcription is taking place, instead of stopping at the mutation, transcription can kind of skip over, jump over the mutation and keep going. So you have a fairly decent protein being made, but again, only a few patients. And then also it requires multiple treatments. And again, the benefits are like marginal, right? Nothing to write home about. Stop codon drugs that prevent breathing through, also there, but also no significant change. So not great. One of the problems with the disease is that they are not really good animal models. Most of the animals that model the disease genetically with mutations in dystrophin don't really get sick to the extent that people. The animals that get really sick are dogs and pigs, but dogs and pigs are not really good genetic model organisms. You can't really run a screen on golden retrievers, right? And so this has been a problem. So we need better systems. Because of that, also, we know very little about dystrophin in a general biology. Like, what is the role of those different isoforms? What is dystrophin doing in all those different tissues? We don't really understand that. do very well right now. And so we need to understand that if we want to understand what happens when it's not there, right? That has led, combine these two things, and that means that we know very little about the progression of the disease. And the treatments that I gave you, they are not addressing the pathophysiology, they're addressing dystrophin itself. So they're trying to deliver a version of the protein and kind of bypassing the disease, right? We also need to look at other tissues. Most of the research is on striated muscles and not really on smooth muscles, for example. There is not much on the neuronal side of things as well. So we need a lot of work needs to be done. And these are things that our lab is currently kind of working on, these different things. So why do we use worms to study a human disease, right? One of the reasons is that worms have been used to study muscles for over 50 years. So there is a lot known about the roles of different genes and proteins in the muscles of worms. A lot of these structures are highly conserved, and I'll show you some of that with dystrophin, including dystrophin and other genes that play a role in the disease. The worms are extremely simple to manipulate genetically. We do this routinely in our lab. And they can model the disease as I'm going to show you today, right? They also they exhibit some of the properties and some of the things that our muscles do. Like you go to the gym, you exercise, your muscles grow. You stop exercising, your muscles atrophy. Worms can model all of these different processes as well. So they are good to study, right? And obviously they are cheap, and we can use them to do different kinds of screens, right? So this is just, we got lots of pictures of dystrophic worms, but this is just a picture of some muscles in a healthy C. elegans. You see labeled here is the actin filaments, part of the sarcomeres in a healthy muscle. These things are under tension, like the strings of a guitar, right? And you can see down here in a dystrophic muscle how those have lost attachment, and now they are kind of collapsed, just kind of like a string. If you cut the string of a guitar, it will go bang, and kind of go back. And this is kind of what you see. There is lots of features about the dystrophic muscles that we can readily observe and quantify. Another thing that we can do with the C. elegans is that we can use genetic reporters. And this is GCAP, a molecule that basically allows the calcium that is present during muscle contractions to bind to this molecule and then convert it to a form that emits light, right? And this is great because that means that we can look at a worm under the microscope and see when a muscle is contracted versus when a muscle is relaxed, and kind of quantify that in a freely moving animal that is going about its business, right? Without electrodes or without any other thing, right? And so here's an example kind of what that looks like when you look at a baby worm. This worm is 1 hour old and it's just hatched. And here in a healthy worm, you see a little bit of calcium in the contracting muscle there, and a little bit of calcium in the contracting muscle there. In a dystrophic worm, you see this worm is brighter, right? And so there's a lot more calcium in the musculature there. As a consequence of that extra calcium, you see that this worm is bendier than the other. So it's hypercontracted, which is a characteristic of patients of the disease as well, right? And one of the take-home messages from this paper from Kylie when she was in the lab is that because you have calcium in the muscle that ought to be relaxing, when you go to contract, you need to contract extra hard in order to create generic propulsion, and that feeds back into the disease, more force, more damage, more contraction, more calcium, right? So it's kind of like a vicious circle. So this is a quantification of the levels of calcium here. And we can do that in a moving animal so that we can see how the calcium levels go from contracted all the way to relaxed, all the way to contracted. And if you remember those two channels, the ryanodine receptor and the Cerca pump, which are basically responsible for calcium coming out and going back in, this suggests that the ryanodine receptor, where the calcium is exiting the muscle, the kinetics there are okay. But the problems are during the pumping the calcium out of the cytoplasm. That's where we see the deficit here, right? All right, so first things, like let's look at the dystrophin gene, which generates lots of proteins, as I mentioned, right? So here you have the human version of dystrophin, and these are models of the functional parts of the functional domains of the protein. The longest of the versions of the protein that is expressed in skeletal muscle is up here, and then there are some of the shorter ones, many of which are expressed in neurons and smooth muscles and so on. And when we joined this picture over the last 50 years, the idea is that C. elegans has a similar array. The gene is about the same size. If you look at the size in amino acids, it's about 10 amino acids off, right? But that's pretty close. And the same domains. And then the idea is that there is only 2 isoforms in dystrophin, okay, of in C. elegans, right? However, when we look at work done with computers analyzing the genome, it suggested that there will be more star size and that there could be actually more isoforms in there, right? And so that's important because that kind of approaches the number and the structure of those isoforms there, right? And so we decided to explore where this was true. And so Mackenzie did this in a couple of different ways. She compared the human and the worm isoforms in terms of the functional domains using SMART and kind of give them a score. And then we also look at the DNA sequence of the isoforms and compare them between the species. And basically the take-home message from these is that, you know, the longest human isoform is most similar to the longest worm isoform and so on, right? So there seems to be a matching, sort of liberal. It's not one, you know, extremely matching, but, you know, the shortest one is closest to the shortest one and the longest one is closest to the longest, okay? So that was enough for us to kind of dig deeper, right? And so in this case, we wanted to know, okay, if there are more than one isoform, maybe we can look and see if they are expressed in different tissues. And we use the regulatory region of these proposed isoforms at this point. And we use that sequence that is highlighted in different colors here. And instead, we put attached to that sequence a fluorophore like GFP so that we could see whether that sequence drove expression of those genes somewhere that we could see, right? That was work done by Adina. And what she found was indeed the different regulatory regions drove expression of this fluorophore in different tissues. The regulatory region upstream of the long isoform was in the striated muscle. You have the short one in neuronal cells and so on, right? So that's cool because it kind of matches what we know from humans. And so the take-home message here is that at least the genes, the human gene, the dystrophin and the worm seem similar in terms of the size, the number of isoforms, the pattern of expression, and so on, okay? And now we wanted to look at these isoforms in these different muscles and see, hey, are these muscles messed up when dystrophin is not there, right? So the worms have a cardiac-like muscle, which is the pharynx shown here, and is used to, this bulb here basically sucks food, and then once it's here, it squeezes it back into the terminal pharynx here, which chews it. It has a tooth in there, and it's chomping on the food, right? So interestingly, the dystrophin is labeled, the protein is labeled in red here, and you should be able to see that there is an increase in the level of the protein in the terminal pharynx compared to the rest of the muscle. And this is cool because this is the part that is doing the hard work of chewing food, right? The other part is just taking a sip, so to speak, right? And so when we look at these muscles in dystrophic animals, we see that indeed there is damage to the terminal part there in the pharynx in dystrophic animals. And if we look at the function of the pharynx, how fast the animals chew food, we also see a defect there, all right? So we're still working on this to do the rescue to see if they get better, right? But we moved on and also looked at smooth muscles. So the worms have different types of smooth muscles, but the uterine muscles that they use to kind of push the eggs out are a set of them. And they also express dystrophin. These muscles kind of get shredded over time. They're only present when the worm is young, and then they kind of stop. And you can see here the uterine muscles in a healthy animal, but they are kind of gone in a dystrophic animal, right? Now, when we put this isoform, this is isoform E of dystrophin, one of the shorter ones that we're tagging here. And when we put that isoform back in dystrophic animals, we saw a recovery of these tissues. And now we can see these uterine muscles again in a dystrophic animal, right? That is also true at the level of the function. So here is a healthy animal egg laying rate versus a dystrophic animal that is impaired. And when we put dystrophin E back in the uterine muscles, we see an improvement. If we rescue a different isoform in other tissues, we don't see an improvement, right? So it's specific to the isoform that is there. Another type of smooth muscle is the enteric muscles. Just like in humans, the worms have smooth muscles and they're used to like poop, basically. And so you have these somatointestinal muscles here that basically that grow like fingers that squeeze the intestine and basically push the food down, right? And then you have the anal depressors that are used to actually defecate. One of the things about muscular dystrophy in patients is that not all muscles are affected the same way. Muscles that generate eccentric force, that is muscles that are generating force while being stretched, all right? So leg muscles and so on, they are particularly under stress and they are particularly affected in the disease. And this is interesting because in worms, these muscles are dorsoventral. And since the worm moves dorsoventrally, these are eccentric contracting muscles, all right? So matching what we will expect will be most affected or most propense to damage, right? Here's what those muscles look in a real worm. And when we tag the protein, as you can see here in red, we saw the highest level of expression of dystrophin in these muscles compared to any other muscle, striated or anything else in these tissues, right? And that kind of matches that idea of eccentric contractions requiring extra care, right? If we look at a dystrophic animal without these muscles, this is the intestine here in a healthy animal. Without the muscles, you see that it's basically distended. There is nothing to squeeze it. And these animals are constipated, right? So the defecation cycle is increased in them compared to healthy animals. And just like with the uterine animals, we can rescue this defect by expressing the correct isoform. We partially rescue the defecation defect in dystrophic animals. But if we rescue the incorrect isoform, then we do not see that. So now we're going to go into the most familiar of the muscles, which are the striated muscles, okay? Let me take a sip of coffee so you guys can breathe. Think. In these animals, in humans, as in worms, our skeletal muscles express the longest isoform of dystrophin, but not only that one, but also some of the short ones. And this is important because most of the mutations that cause muscular dystrophy selectively target the longer isoforms. So most of the patients have mutations that take out the longer isoforms, but not the shorter isoforms, okay? And despite the fact that those shorter isoforms are also expressed in striated muscles. So that was kind of cool when we saw that striated muscles also express both longer isoforms and shorter isoforms, and you can see them in the developing embryos here. And we saw a closer zoom in that the level of expression of the different isoforms here labeled with different fluorophore was different from cell to cell. And this is cool because what we can do is that you can convince a student in your lab to dissolve these animals until they are a soup of cells. Thank you, Mackenzie. And then you can convince another student, Danny, to pass these through a FACS sorter where the cells can be sorted based on their size and on the different levels of different fluorophores, right? And so these will allow us in future to kind of quantify gene expression and protein expression on muscles sorted by the level of expression of different isoforms, for example, right? And tell us, how do you control that expression? Another thing that we saw was that the level of isoforms that these muscles express was experience dependent. And so we made animals tunnel through agar of different densities, and we saw a change in the amount and in the ratio of the expression of different isoforms. And now this is promising because this says that the animal has a way to modulate one isoform versus another, right? And so if we can trick the animal to upregulate the shorter isoforms, maybe we can trick patients to express short isoforms and somehow be improved, right? So how do those worms know that they need to modulate this? One of our hypotheses is that maybe some of the mechanoreceptors that they express in muscles may play a role. And you might be surprised, like I was, to learn that our skeletal muscles express over half a dozen different kinds of mechanoreceptors, right? Some of what they do is understood, but most is not. What are they doing there, and why do we have them, right? And so in this case, this is work by Adina, we use RNA interference to silence the expression of a mechanoreceptor. And when we look at the ratio of these dystrophic isoforms, we saw that that could be altered, right? Another thing that we did was we went back to that gene diagram and we look at that regulatory region in front of the short isoform, and we look for binding sites for transcription factors that may be modulating it, right? And so when we found some that were shared with humans, okay, so it's a quantification. We use RNA interference to silence this transcription factor, and we saw a change in the level of expression of that particular isoform, right? And it was that particular isoform because then when we measured the ratio of that isoform to the other, then we saw that there was a difference. Okay, so from here, the take-home message is that dystrophin has different isoforms. They are expressed in different tissues, and they lead to tissue-specific deficits. And I'll add another one to that, is that they are plastically modulated, right? You can change the expression of these isoforms with experience or whatever, you know. So the next part is going into, now that we have this system that is similar, close to how the human gene works and similar defects, similar function, we want to use it to try and look at the disease and try to answer this question of what is responsible for this difference in calcium. Because before the muscles break, the calcium goes up. And maybe if we can identify how calcium becomes dysregulated, we could target that as a treatment and delay the onset of the disease, for example, right? So these are, as I mentioned before, 1 hour old larvae. And as you can see, they are brighter in the dystrophic animals, showing that they have higher levels of calcium. And that is quantified here, right? And so it's like, okay, even a 1 hour old baby is already brighter. How about the embryos? Turns out that the embryos are also higher in calcium, right? Okay, so maybe they are higher in calcium. There could be many reasons for this, right? Maybe they are moving more, maybe they are older, they're developmentally more advanced or something, they're born later. And so we wanted to investigate this, right? And so we look at the hatching patterns in these animals, and what we found is that the dystrophic animals actually do hatch a lot later compared to healthy animals, right? Like these are synchronized here, and they end up hatching a lot later. Now, there's many reasons for this could happen. So we got 3 alternative hypotheses here. One of them could be that there's just developmental delay. The animals simply take longer to arrive at the point at which they hatch. Another one was maybe they are laid early. The mothers have muscular dystrophy. So maybe they're supposed to be laying the eggs at 150 minutes post-fertilization. If she lays those eggs earlier than that, the egg will have to cook outside of the animal longer, right? And so maybe that's why. And the third idea would be maybe these animals are struggling to hatch, and maybe they are not strong enough to break the eggshell and get out, right? So we set out, because C. elegans kind of develop outside of the animal, we can see the different stages, and people make a nice living out of looking at this development. the eggs are laid at 150 minutes post-fertilization, and they hatch 840 minutes. And this is the heroic part of the talk. My students had to sit there and take pictures of this thing over and over and over again to kind of get an idea, right? So the first thing we did is, are these things developing slower, right? And so we look at eggs starting at 150 minutes and see when they arrive at this pretzel stage that they look like a pretzel, and there was no delay there, okay? So they develop at the same speed as a wild type animal, okay? Maybe the mothers are laying the eggs too soon. And so we look inside the moms and you can see the eggs. And we look at the eggs and see what stage are those eggs. Are they all at the gastrula stage, or are they all earlier than that? And let's quantify that. And when we did, we saw the opposite of what we expected or what we predicted, which is not only were the dystrophic mothers not have extra early embryos, they have late embryos. And they were laying eggs that were older than they should be, right? So that was not an explanation for why these animals are hatching late. And that took us to the third hypothesis, which is maybe these animals are weaker, and that's why they struggle. So we use RNA interference to knock down a gene that is used to make the protein that makes the eggs stiff, and therefore, by doing that, make it easier to break from the babies, right? Basically make the eggshell softer and easier to punch through, right? And that's what it looks like. When the eggshell is there, it looks like that. If it's not there, it loses that structure. It just becomes like a ball, right? And lo and behold, when we did that, there was no effect on wild type animals, but it basically completely rescued the hatching time on the dystrophic animals. So that hatching delay was explained by that strength, that lack of strength of the animals to hatch, basically, right? So at this point, we say like, okay, this is over. Now we have the reason, and therefore, normally what you expect now is that while these animals will be brighter than wild types, if you look at the GCAM signal, these ones now aren't, because they're now hatching normally, right? So they should not be struggling anymore. Do you think that's what happened? No, it's not what happened. So what we saw instead was that there was an improvement in these animals. They were significantly dimmer than the controls, but they were still brighter than wild types. So even though we made it easier for them to hatch, their calcium was already elevated still in those animals. So that means the elevation in calcium is not hatching related, basically, right? And so at this point, I had to have a sit down with my students to tell them, you had to go and watch the whole thing, from beginning to end, and take a picture every 30 minutes, please, or 2 pictures. And so what we did, and I'm just showing you part of this, is we, in this case, we use a drug that paralyzes the worm. And so we hypothesized if we paralyze the embryos, they can't move. And therefore, there could be no mechanical damage to the muscle, and therefore the calcium won't get increased, right? And that was true later when the animals are about to hatch. We can put this drug, it's called tubocurarine. It's a muscle relaxant. And if you put that drug in there, the embryos inside the egg, their movement is decreased dramatically, right? Early on, this is before they have contractile ability, nothing happens, right? You don't expect that to work. And then we look at the calcium levels in these embryos. And as we expected, we saw a drop in the calcium level later on. But surprisingly, we saw that the calcium levels or the GCAM signal in the dystrophic embryos was significantly elevated already before that muscle was able to physically contract. And using this drug, as you will expect, it had no effect on those muscles because they're non-contractile, right? But this is interesting because this is showing that even before the muscle has developed enough to generate contractions, the calcium is already elevated. So at that point, we need to use what C. elegans is good for, which is genetics, to do a screen. And there are many genes important in muscle function Some are important for ions or letting calcium into the cell, the dystrophin and the dystroglycan complex, the contractile machinery, the endoplasmic reticulum, and the renin receptor and the ATPase pump. So all of those play a role, and we silence each and every one of those and look for changes that may signal an effect, right? But the moneymaker, or the one that really was interesting, was this particular calcium pump, MC1 or PMC1 in humans. And what we saw there was that this is a pump that is constantly pumping calcium out of the cell. It's kind of running, it's like a sump pump in your basement. As long as there is water in there, it just kind of pumps out, right? And so what we saw here is that when we silenced that pump in dystrophic animals, there was no change. But when we silenced it in wild-type animals, in healthy animals, there was a significant increase, right? There was another similar pump, PMC2, also around there. If we silence that one, we saw an effect both in wild-type and dystrophy. So what is this saying? This is saying that dialing down MC1 in healthy animals make them look more dystrophic. But silencing that in dystrophic animals doesn't, right? Then we went on and actually found that in the worm, that pump is actually downregulated in dystrophic muscles, right? And so it seems like the reason dystrophic muscles have elevated calcium, at least early on, is because this pump is not being expressed at normal levels. But the question is, okay, that's in worms, but we want to cure humans, not worms, right? So what happens in humans? So here are some muscle cells, striated muscle cells from human patients. These are healthy cells here, and these are dystrophic cells here. And this is labeling actin and dystrophin, the parts of the muscle that contractile bits and the structural bits, and this is the merge to see that this is there. And we can grow them in the lab all the way to the myotubes, and we can stimulate them and so on. And so we're using them to kind of validate the things that we have been discovering in worms. And one of the things that we saw is that even early cells, like 24 hours, myoblasts, already show an increase in calcium in the dystrophic cells. So again, these are non-contractile cells already elevated in there, okay? So that matches the worm. We can let those cells develop into myotubes, and then with Wolfgang's lab's help, Danny can go and stimulate them electrically. And you can see here the calcium transients in a healthy cell in black. You see these 3 spikes are the same size here, and also here. However, the dystrophic spikes have a greater amplitude, but then they go down, they deplete later on, right? And we can quantify that here. We also see that this is normalized here, but the dystrophic cells are also much brighter than the wild-type cells. That's quantified right there. So, okay, so we're seeing the same thing as we see in the worm. How about this MCA1 pump, right? So the human version of that is a PMC1 pump, the PMC1 protein, and we use antibody staining to label that in wild-type cells, myoblast, and then in wild-type and dystrophic myoblast. And you can see that there is a brighter signal here, meaning there is more protein in the wild-type than in the dystrophic. Now, that's at the level of the protein, but Danny also did this with qPCR to measure gene expression. So you have the RNA levels are also graded in the wild type than in the dystrophic as well, right? So this is showing the same result that we saw in the worms, right? So when, what am I looking at here? Okay, so this is another quantification of that. I'm gonna skip through. Now, we then use a blocker rather than downregulate we downregulated with RNAi, and we saw the same result. And now we use a blocker to block the pump. And again, we see that when we block the pump in dystrophic animals, there is not really a change. But when we block the pump in healthy animals, we see a change. And back here, it was downregulation with RNA interference. And we basically see the same thing in the human cells. So every time that we do that, and this is just showing you that this is darker than this, but this is not so much darker than that, right? So that's what this is quantifying. So either blocking the pump or downregulating the pump has the same effect. And if we let these muscles develop to the myotube stage, and then we stimulate them again, this is your data, Danny, I just graphed it. Now you can see this is the wild type response without the blocker, but if you block the PNC1 pump, this is the response much increased, right? In contrast, in the dystrophic animal, there is some effect, but it's much reduced compared to the wild type, all right? So in the future, we're going to continue to look at that calcium and figure out what's happening there. We are trying to build a rig to be able to like measure the force coming out of these muscles so that we can not just measure calcium transients, but also force output from the myotubes. We're looking at the corticosteroid to see how they work and whether they can be improved. Continuing to work on the different tissues and different isoforms, all right? And so without that, I'm going to skip and go to the people who made this possible. So this is the folks in the lab. Some of them graduated, and the ones in reds are the data that I presented today. You know, if you know somebody who wants to join our lab and work on their muscles, tell them to give us a call. And with that, I'll be happy to take any questions. Thank you. Yeah. Yeah, so that was one of the things that we were interested in learning, because I work with patients with DMD when I was an undergrad, and I was living with them, like, you know, to take care of them. And they do all kinds of treatment because nobody knows what works, right? And so they try everything. And they try physiotherapy, they try aquatherapy, they try everything that they can think of. The problem with dystrophic muscles is that your muscle, the mind, if I go to the gym and I pull something like I did recently, slowly at this age, and better when you're young, you get better from that. In the case of dystrophic muscles, they do not. Like if you have muscular dystrophy and you do something to your muscle, that is the new you. It won't get better ever, right? And so what I saw these patients doing is that they were trading what little health their muscles had left for a chance to maybe get better. But in most cases, they go worse, irreparably worse, right? And so our first paper on this showed that that was the case with worms too. But yeah, that's why we wanted to know, is there any kind of activity that you can prescribe that might improve the muscle? In the literature, anything that people have done, they measure one variable, you know, and they're like, oh yeah, this thing, we measure one thing and that's better. But they don't measure all the different metrics, and they don't go back and look. And in reality, that's never panned out. In muscular dystrophy, it's a horrible field because all these treatments that even people that are FDA approved, 1.5% improvement over base, right? Like nothing, you know, and millions of dollars going into that. So yeah. Yeah. That is definitely something that we are working on. The R15 that I got reviewed recently proposes to actually look at the different muscle types within the same individual worm, so that we can track the disease in the striated muscle, in the smooth muscle, and in the cardiac muscle in an individual, and see, does the disease affect all the muscles the same way to the same extent That's the treatment. Do the corticosteroids make them all better, or does it make some better and some worse? And so I'm hoping that we can start looking at closer, in that respect. The other thing that we might be able to do is use this fact sorter to kind of like use fluorophore reporters to kind of separate different cells even, or different worms based on the expression levels or the calcium levels, and then do an analysis on RNA or at the protein level. Yep.
Yes, one of the bigger questions is like the pump, the PMCA pump, and its co-regulation with dystrophin, right? Somehow, like the dystrophin is gone, the pump is
Down.
down.
Oh,
Is there a way to like in patients potentially, like in animals that already have damage, like in celiacs,
yeah.
to like increase dystrophin, the wrong dystrophin expression to see whether the pump.
Yeah, that is also in the grant that we propose. That we are proposing to use, instead of using the full length dystrophin, use some of the isoforms to rescue different tissues. And so that would be an idea, because one of the things that he's alluding to is that those are 24-hour-old myoblasts. They're very early on. Dystrophin takes 14 hours to transcribe, right? Like to make the protein, the full length, right? So there is not enough time in a myoblast to make a full-length dystrophin, package it, send it, have it sit there, regulate 3 genes, the chain of events doesn't allow for that kind of thing to happen. So something else must be happening that is being inherited in this muscle. Some other regulator must play a role. And so one of the things, like with the RNA-seq, for example, we want to do that, but also by introducing some of the shorter dystrophin, maybe the short dystrophin that is very small, that is also transcribing muscles, maybe that is crucial for the PNC pump, and that will allow for that time course to be reconciled, right? So yeah, we'll definitely want to do that. One of the things that Danny could do, is measure the expression of those shorter isoforms in the cells that we have right now, right? Are those being expressed in those animals? Yeah.
You started with a comment about phospholipid membranes being fragile. Is there any role for dystrophin in protecting the phospholipid membranes of the sarcoplasmic reticulum?
That's a great question. I don't know the answer to that question. I don't know that many people know. We haven't seen it, but we haven't really stained the sarcoplasmic reticulum. But we do have worms that have, Mackenzie, I'm looking at you, that have blue camp in the sarcoplasmic reticulum. And we do have a worm that has tagged dystrophin. So you could look in those animals and see if the blue and the red are co-localized, and that would, because that would be a great mechanism for leading to that, dystrophin dysregulation. If the ER, which by the way, in muscles, the endoplasmic reticulum is like the whole surface of the cell is covering this thing. It's not just like a little pancake sitting on one corner. It's all over the place. So it would make sense, that would make sense to look at. Yeah, we need to go because there is another class. Thank you, everybody.
See you guys online.
Bye, friends.
Bye. Bye.
Thank you for coming.
Bye.
Oh, you're welcome.
Good talk, Andres.
Thank you.